Hello everyone, welcome to this special lecture series organized by BK Modi Government Pharmacy College for forthcoming GPSC examination. Myself, moridhawal, Assistant Professor at BK Modi Government Pharmacy College. First of all, I would like to thank everyone, those who have watched our series and appreciated it. Thank you very much. Second thing, I would also like to tell that please share your valuable feedbacks on the email ID that is given. Your feedbacks are very valuable. It will help us in making lectures more cogent, more student oriented. And obviously, as is to human is to err. So if you think that we have made any mistake in description or in some factual things, then please share also. We will also try to correct it, and we will try to make this lecture series as student oriented as possible. So after this introduction, let us get back to our topic. So in our previous session, we have gone through the questions which were asked in an examination related to microbiology and biotechnology. So that we are going to continue in this session also. So let us get back to questions and answers to that with explanation. Okay, so one of the question which was asked in was that which of the following exam is not restriction endonuclease. So in our last lecture we have discussed the function of the restriction endonuclease enzyme. So we know restriction endonuclease enzyme is one of the defense mechanism that bacteria is having. So whenever virus infects the bacteria, it is the tool by which they can cleave the viral DNA and protect themselves. But you see in this question, one of the option that was given was the DNA ligase enzyme. So DNA ligase enzyme, generally it is one of the important component of the DNA replication process. So I thought that because this option was given, it is important to understand the function of the DNA ligase enzyme. And DNA ligase enzyme, because it's a part of the DNA replication process, I think it's worth looking at the entire process, because this process, you can see it's a very ubiquitous process, and there are lots of players which are participating in this process. So questions might be asked from this portion. So we all know what is replication process basically. So replication process basically is nothing but it's a process by which a DNA strand can be synthesized from the parent DNA strand. So parent DNA strand means the DNA strand which is already exist, and by this process we are copying the that information into the new DNA strand. So basically it's the DNA replication process. So this replication process involves, as you can see in this list, it involves lots of players. Now one of the key characteristic of the DNA, its success as a genetic material, is that its ability to replicate itself. So DNA has a unique ability to replicate itself. So as you can see in the list, there are lots of enzymes involved in this process. So let us see one by one how this replication process proceeds. So as we discussed in our last time lecture, we know that DNA basically is a double helix. There are two strands which are attached with each other by hydrogen bond. So one strand is going in 5' to 3' direction, and second is going in a opposite direction. So it's a two-strand DNA. Now to first start the replication process, first thing that is we required to do is that we have to open this coil, or we have to unwind the DNA. We have to separate two strands. So first thing the DNA replication process start is the opening of the two strand. It's the separation of the two strand. Now second thing, the DNA replication process doesn't start randomly. It doesn't start anywhere. Only the specific sequence of the DNA which is recognized by the enzyme, and from where the replication process start, and that is known as a replication fork. So replication fork is the specific sequence of the DNA from which the replication process start. So the first enzyme which participate in the replication process is known as a DNA helicase. So DNA helicase is an enzyme which this enzyme is responsible for separating the two strands of the DNA. So this enzyme basically separates the two strands of the DNA, which is the first step in the replication process. So as you can see in this figure, two strands first are separated. So DNA helicase is the enzyme which separates the two strands. Now once these two strands are separated, these strands always have a capacity to again join with each other. So to make sure that once these strands are separated, they won't attach with each other again, a protein called single strand binding protein, it binds with this single strand of the DNA and stabilizes them. It means it will not allow this strand to again join with this one, because we know both are joined by hydrogen bond, so they can easily reattach themselves. So to prevent that, second player in this DNA replication process is the single strand binding protein, which binds with this separated strand, and it will not allow this strand to again join with the complementary strand. So that is the single strand binding protein. That is the second. Third one participant in this process is the enzyme that is known as a topoisomerase or DNA gyrase. So what basically topoisomerase and DNA gyrase does is that, as we know, DNA is a double helix molecule. So what happens is that if we open this strand from the middle, the remaining portion of the DNA strand, it experiences the extra pressure. It is known as a negative supercoiling. So it experiences the extra pressure. So this extra pressure, which is generated due to opening of the two strands, it is released by enzyme called topoisomerase. So generally two subtypes of the enzymes are used. In a prokaryotes and eukaryotic, two different type of the enzyme, both are having the same function, but they are having the different names is given. So in a prokaryotic cell, the enzyme is called topoisomerase, and in a eukaryotic, especially in a bacteria, the enzyme which is doing the same function, it is known as a DNA gyrase enzyme. So these two enzymes, both what they do, they relaxes the DNA because of the supercoiling because of the DNA supercoiled nature. Now the third participant in the process is called RNA primase enzyme. What is its function? Let us see. So what happens with this process is that we know that once the two strands are separated, now to replicate this strand or to copy this strand, we required enzyme called DNA polymerase. So we know DNA polymerase is the main enzyme which recognize a nucleotide one by one, and they will add the nucleotide molecule. But what is problem with this DNA polymerase enzyme is that the DNA polymerase enzyme cannot bind with the single strand. To start the replication process, it can only recognize the double strand DNA. So what happens in the DNA replication process is that initially when two strands are separated, in one strand, this is first strand of the DNA. Now because this single strand is not recognized by DNA polymerase, an enzyme called RNA primer, it will make short RNA sequence. So remember, it's a RNA sequence. But this RNA sequence is complementary to this DNA, right? Complementary, we know that how hydrogen and nitrogen bases are attached with each other. We are not going in that because we have already discussed in our previous lecture. So complementary base pairing, we know a purine is always joined with the pyrimidine. So what happens that according to the sequence which is given on this DNA, a short sequence of the RNA is formed, and this is known as a RNA primer. So RNA primer is a starting point. Now because now this has become double strand, this double strand DNA this double strand is recognized by the DNA polymerase enzyme, and this DNA polymerase enzyme will start to add the nucleotide molecule from 5' to 3' direction. Right? So the question can be asked is that new strand of the DNA is always synthesized from 5' to 3'. It's the only direction where it gets synthesized. So what happens now? Once it is the starting point DNA polymerase having, it recognizes this starting point, and once it's recognized, then one by one it will start to read the nucleotide molecule given on a parent strand, and according to that it will start to add complementary base pair, right? Complementary base pair, we know adenine is joined with the thymine and guanine is for cytosine, right? So it's a complementary base pairing it is having, and these it will read one by one and it will start to add nucleotide molecule. So the third players in the DNA this polymerization reaction is the enzyme DNA polymerase, which is the main enzyme which reads the nucleotide sequence, and based on the nucleotide sequence it's going to add the new nucleotide. So RNA primase enzyme, it gives the starting point. Once the starting point is recognized by DNA polymerase enzyme, it will start to add nucleotide molecule. Now same thing, because as we discussed that newly synthesized strand is always synthesized from 5' to 3'. One strand where the 5' end is open, it's going to synthesize continuously. As you can see in this figure here, there is a continuous 3' end is here. So it synthesizes continuously. But if we think about the opposite strand, here it's not the case. Here you can see the 5' is here in opposite direction and 3' is here. So opposite strand, it cannot synthesize continuously. It synthesizes in the fragments, and these fragments are known as a Okazaki fragment. And here also this strand where there is a continuous synthesis, it is known as a leading strand, and opposite strand is known as a lagging strand. So this opposite strand where the DNA is synthesized in a patches, in a small fragments, they are known as a lagging strand, and these fragments are known as a Okazaki fragment. So here also you can see these are the RNA primer. They are the starting point. This is recognized by the DNA polymerase enzyme. As you can see, this DNA polymerase enzyme recognize it and it will start to add nucleotide molecule. So RNA primer is giving the starting point and DNA polymerase is extending the chain. So once this RNA primer started to giving the starting point and this synthesize, in this opposite strand what is happening? It's not completely DNA, right? It's a combination of, because there are small patches of the RNA and DNA, and obviously RNA is not acceptable in a DNA sequence. So what happened is that we have to do that this RNA is required to remove, because the only purpose of this RNA primer was to give starting point. Once it is over, then it is required to remove. So that function is performed by enzyme called RNase. So what this RNase does, it will remove the RNA primer. Now once the RNA is removed, whatever remaining portion is that, again that portion is filled by RNA polymerase enzyme. So now the RNA is removed, whatever patches are remaining where there is no nucleotide, it is again complete by RNA polymerase enzyme. And because now this RNA polymerase, DNA polymerase has added nucleotide in two different parts. First it was here, and second in a places where RNA was removed. Now these two separate strands of the DNA is required to be joined, and that joining is performed by the enzyme called RNA ligase. So RNA ligase, it joins the Okazaki fragment by phosphodiesterase bond, right? What is phosphodiesterase bond? We have already discussed in a next lecture, so we are not going in a detail. So as you can see, the entire process, entire process is completed by various participants. DNA helicase first separate two strands, single strand binding protein. It stabilizes the strands of the DNA and preventing from again joining with each other. Tropoisomerase and DNA gyrase in bacteria, tropoisomerase in eukaryotic cell, they relaxes the DNA, supercoiled DNA. RNA primer, that gives the starting point, so replication process can start. The main enzyme which participate is the DNA polymerase enzyme, and as you can see the word is the polymerase. So basically what they do, they read the sequence and according to that they will add the complementary base. Now once this complementary base pair are added, we require to remove the RNA primer. So RNA primer, the starting point, it is removed by RNase and the DNA ligase joined the enzyme. Now another another enzyme which is particularly special for or particularly exclusive for the eukaryotic cell is the telomerase. So what is basically telomerase? So telomerase, as you know, in a eukaryotic cell the chromosomes are linear. So this end portion, to replicate this end portion, we require a special enzyme called telomerase. So telomerase is a special enzyme which is required to replicate this end portion. Now how exactly telomerase works, why it is required in a eukaryotic cell only. Actually, it's out of scope of this discussion. If someone is interested, you can go in a YouTube, you can see it. Because if I explain this telomerase enzyme, its function, it's another lecture. So just remember that telomerase is an enzyme which is required to aid at the end of the chromosome. And because this is eukaryotic chromosomes are linear, they require this telomerase enzyme to replicate, to complete the replication of the end portion. So this is the replication process for the replication process involved in a right. So it's the right. Okay, so as you can see in this figure, the various players and how the entire process proceeds. Now, second thing, question which I was there, which is related to, not exactly related to replication, but it is related to conversion from the DNA to protein. So, as we know, what is the function of the DNA? DNA is the molecule which basically contains information, and this information is in terms of protein synthesis. Ultimately, we know the DNA converts into DNA, it forms RNA, and from RNA we have this various protein, right? So DNA is basically containing information to make the protein, and this process of the from reading the information which is given in a DNA and converting into protein, broadly it is known as a protein synthesis, but we know it's a two-part process, right? First is known as a transcription process, and second is known as a translation. So transcription, what is transcription? Basically, we read the sequence which is given in a DNA and convert into RNA. So exact process we are not going, we know a messenger RNA is formed, then tRNA brings the amino acid, and because it's a very basic things, so we are not going in a detail. So basically, DNA is converted into messenger RNA, this messenger RNA then converts into protein. So this is known as a transcription, and then there is known as a translation process. So what happened, one of the key difference between the prokaryotic cell and eukaryotic cell, apart from whatever difference that we have discussed in our previous lecture, another key difference between prokaryotic cell and eukaryotic cell it comes is that what happens once the transcription phase is over. So as you know, what is basically transcription is DNA is act as a template, and from that enzyme called DNA dependent RNA polymerase, right? So enzyme called RNA, or it is simply in simple terms, it is also known as RNA polymerase. So RNA polymerase enzyme read the sequence given in the DNA and they form the RNA, right? So this is the transcription process. So what happens in the, what is the one of the major difference between prokaryotic cell and a eukaryotic cell is that prokaryotic, you can simply think the bacteria. So what happens is that in the prokaryotic cell, once your DNA is transcribed into RNA, this RNA directly goes to ribosome, and then it starts the translation process. So whatever RNA is formed, it directly goes to the ribosomes, right? And where the transcription process or translation process start. Now what happens in a eukaryote? It is not the case. What happens in a eukaryote? Initially, whatever RNA is formed due to transcription process, this RNA is known as an premature mRNA. Premature, it's not complete RNA. It's a RNA, but this RNA is not ready-made use. It cannot directly go to the ribosome and start the translation process. What happens is that between that means between the formation of the new RNA and before it reaches to the ribosome where the translation process starts, there are some processes that is required to perform on this mRNA strand, and that is known as a post-transcriptional modification. So you can see the term. Post means after, transcriptional, that means once your transcription is over. That means once your RNA is formed from DNA. Remember, only it is very specialist, or it is a special class in the eukaryotic cell only. So we can say it's a unique feature of the eukaryotic cell where the RNA which was formed from the DNA is processed. It is performed some reaction and then it is converted into mature RNA. And that mature RNA reaches to the ribosome and then the translation start. So the process of converting this formed RNA into working RNA. So this was the RNA which was formed and this is the RNA which is a working RNA. So to convert this formed RNA into mature functional RNA, the process is known as a post-transcriptional modification. So it's a modification process when it is happening post-transcription. So when transcription is over, this process happens. So basically what happened in this process, obviously this post-transcriptional modification is a very lengthy process and obviously it involves lots of players. So it's not possible to cover all the aspect of this process in this lecture because this lecture is expected to just give you the idea about questions, not the entire topic. Though I am going to explain some part of it. So let us see in the next figure. So as you see, this is premature mRNA. So when mRNA is formed, it is something like this. Now this is the mRNA sequence only. What is the key characteristic of this mRNA sequence formed in the eukaryotic cell is that there are some portion of the messenger RNA that are not applicable, that are not taking part in a protein synthesis process. So once your mRNA is formed, this portion which is not expected to participate in any protein synthesis process, that is required to be removed. And that process of removing this intermediate portion which are not expected to participate in a protein synthesis process is known as a splicing process. Splice means to cut, right? So it's a splicing process. So entire mRNA sequence can be divided into two parts. The portion which are expected to participate in the protein synthesis process and the portion which are not expected to participate. So the portion which are expected to participate in a protein synthesis process is known as an exon. Exon stood for expressing part. So this part is going to express and it's going to form protein. So you can see the functional part is known as a exon. And the portion which is expected to remove, which are not expected to part in protein synthesis process, and that is why it is required to remove, it is known as an intron. So you can say the entire premature mRNA, it is composed of exon, expressing part, and intron, which is interrupting part, which is not expected to form protein. So what happened, as you can see in this process, basically what is splicing or what is the post-transcriptional modification is the removal of this intron portion. So obviously it's a lengthy chemical process, various enzyme take participate, but basically what happens, if we take the overview, this interrupting portion is removed and the portion which is expected to participate in a protein synthesis process, they are joined and forms the mature mRNA. So now this is mature mRNA, this is a functional one, this mRNA reaches to ribosome and then as you expect the protein synthesis start. So this entire process where premature mRNA is converted into functional RNA is known as a post-transcriptional modification, and this is a unique feature of the eukaryotic cell. So if we read our question again, what was the question? Question was that the post-transcriptional modification is absent in. So absent in, as I have explained, this is the unique feature of the eukaryotic cell. So obviously bacteria are the prokaryotic cell, so you don't find these kind of the post-transcriptional modification in a bacteria. So answer is bacteria. All the eukaryotic cell you find this post-transcriptional modification. So let us see next question. Now same way, it's not a question but it is linked to whatever we have explained. As one unique feature of the eukaryotic cell is the post-transcriptional modification. Another unique feature of the prokaryotic cell is something called post-translational modification. So here also you can understand it's a modification process, but when it is happening, once your translation is over. That means once your mRNA is read and converted into amino acid sequence, right? We know amino acids, how protein has a primary structure, secondary, ternary and quaternary structure. Once your protein is formed, this protein is processed. So in a eukaryotic cell, whatever protein is formed, these proteins are attached with some functional group. It can be sometimes attached with some carbohydrate molecule, sometimes it is attached with some lipidic molecule. So once your protein is synthesized, then whatever functional modification is performed on this protein, it is known as a post-translational modification. Generally, that is happening as a Golgi apparatus and endoplasmic reticulum. So you can see another, you can see the examples of post-translational modification. Like post-transcriptional modification, post-translation modification is also the unique feature of the prokaryotic cell, eukaryotic cell only. So you can see over here, this is the protein which is formed. Now this protein can have a lots of modification. Sometimes methyl group is attached, acetyl group is attached, depending on what function this protein is expected to perform, these functional groups are attached. So these are the examples of post-translational modification. So once your protein is formed, whatever modification is performed, that are known as a post-translational modification. And as the post-transcriptional modification is a unique feature with the eukaryotic cell, same is the post-translational modification. So if the question is asked that which of the following organism doesn't have the post-translational modification, the answer remains the same, that is the bacteria. So, or the prokaryotic cell don't have all this modification, they are very simple organisms. All these modifications are a feature of eukaryotic cell only. Okay, then the next question which was asked is regarding the PCR. So polymerization chain reaction. The question was that the first step in the PCR, which is the first step in a PCR. So let us see PCR, the entire process of the PCR and how they proceed, because obviously it's an important question. If we see the PCR technique, basically what is PCR? PCR is nothing but if we understand in simple terms, is the DNA replication process which is happening outside of the cell. It is happening in vitro, and that is why PCR also known as an in vitro DNA replication process. The purpose remains the same. We want to make lots of copies of DNA. The aim of the PCR is to make copy. But here the process is happening not inside the cell, but it is happening outside of the cell. This process was basically invented by two scientists, Kerry Mullis and Michael Smith. They invented it because PCR is a very popular process. This question is also the part of your GK. They can expect this kind of the popular process to be remembered. So it was invented by two American scientists, Kerry Mullis and the Michael Smith. So as I said, what is basically PCR? PCR is nothing but it's a replication process. So how this thought came into their mind? Basically what is happening in a replication process, as we discussed in a replication process, is the process where the two strands are separated and then mainly the DNA polymerase enzyme which recognize the strand, one strand, read it, right? So what is reading? It reads the sequence of amino acid. It reads the sequence of amino acid, and based on this sequence, it will add the complementary base pair. That is what happening in vivo when the DNA is replicating inside the cell. Then if this process is performed by enzyme, and basically this process required the separate DNA, then can we perform it outside if we provide all the essential ingredient to have the replication process. We discussed what are the essential ingredients for the replication process, right? So if we provide everything outside of the cell, is it possible to replicate the DNA? And that is what the polymerase chain reaction is about. It's about the replication process happening outside of the cell. One of the major difference between the natural process and this PCR reaction is that it's the first step. As you remember, in the DNA polymerase enzyme, there was an enzyme called DNA helicase. The enzyme which was responsible for separating the two strands. Now, rather than using this enzyme, PCR reaction, it uses the heat as a medium to separate the two strands. So basically what happened, in our DNA structure we have discussed, there are 3 bonds in the DNA, phosphodiesterase, glycosidic linkage and the hydrogen bond. And because hydrogen bonds are the weakest, so what is the expectation is that if we raise the temperature of the DNA strand, and because the hydrogen bonds are weakest, there are chances when the temperature is raised, the hydrogen bond will be broken down and two strands will be separated. And that is what happened in the PCR. So PCR is a reaction which is controlled by thermal heating cycling. Here thermal cycling, it controls the separation of the DNA and joining of the DNA. Otherwise the entire process remained pretty much similar to the DNA replication process that we have already discussed. So what are the different steps in the DNA replication process? As I have explained, that DNA replication process basically it deals or it depends on the thermal heating cycling. So what happens is that this is the DNA replication process. So when the temperature is raised, these two strands which are joined by the hydrogen bond breaks down and we have the separate strand. So now remember the DNA replication process. If we start the DNA replication process, what we require? We have the separate strand. First thing what we require is the primer. Remember the one of the major limitation for DNA polymerase cannot recognize the single strand. So what required? This RNA is going, this primer is going to join and this will act as a starting point. So once we have the starting point, then the enzyme DNA polymerase will recognize this double strand and it will start adding the nucleotide molecule from 5 prime to 3 prime. So based on this description, you can see what are the essential part of the replication, this polymerization process. First, heating process. You require heat to separate. Second, you require primer as a starting point. Then you require DNA polymerase and you require nucleotide molecules which can be attached. So entire process is controlled by cycle of heating. So what happened? First step in the replication process is the denaturation process. What is denaturation? Now denaturation here doesn't mean your DNA is degraded. It means when you raise the temperature around 94 to 98 degree temperature, two strands are separated. So the first thing is that separation of the two strands. Now in a second strand, what we required is that we required RNA primer to join, and obviously if the temperature remains 98 degree, they cannot join, because as we have seen, when the temperature reaches 94, 98, two strands are separated. So what is the second step is that the temperature is reduced from 94, 98 to 50 to 65. And when the temperature reaches 50 to 65, there your RNA primer, it will attach with this DNA, and that is known as the annealing step. So here two things are there. In a PCR, in natural replication process, that is in the natural replication process, where the naturally in vivo is happening, this starting point was given by RNA, because RNA primer enzyme was doing. Generally in this DNA PCR process, rather than using RNA, we use DNA only. So this is DNA primer. If you remember, the difference between the DNA and RNA is nothing. When it comes to single strand, the only difference is the one nitrogen base. So in the PCR process, this starting point is generally given by DNA primer. Function remains the same. Once your DNA primer is joined, it's a starting point which is recognized by DNA polymerase enzyme and the process start. But what I am trying to emphasize is the difference. In natural replication process, this is RNA primer. In a PCR, this is a DNA. Function remains the same, giving the starting point. So once the temperature is around 50-60 degree temperature, at that time this DNA primer, or in general we get primer, joins with this strand and gives the starting point. Now then, once the temperature is that, once your DNA primer or primer is joined with the separated strand, now it's time for DNA polymerase to act. Then temperature is further reduced as 40 degree temperature. So as we know, most of the DNA polymerase in a eukaryotic cells, they have the optimum temperature of around 37. So temperature is further reduced to 40-degree temperature. At a 40-degree temperature, DNA polymerase enzyme joined with here, and it will start to add nucleotide molecule. So you can see, once it start to add nucleotide molecule, you have 2 copy. Now again, the 2 copies of the DNA are not enough. We require more copy. Then what we have to do? Again, we have to go the first step. So again, the temperature is raised to 90 to 94. So what is the entire process? First, denaturation, that is separation of the DNA around 90-degree temperature. Then after annealing, annealing means joining of primer with the DNA. So that is happening around 50 to 65-degree temperature. Once it is there, again the next step is that once the 2 strands are ready for replication process, the DNA polymerase enzyme bind over here and they will start the replication process. But it is around 42 degrees Celsius, and that is known as the extension. So denaturation, annealing and extension or elongation are the 3 steps in a polymerization reaction. And once one cycle is over, again the cycle shift to the first step. Now if you see this process and this process is that one of the key part in this process is that we require to go 40-degree temperature to make sure that the DNA polymerase enzyme work, because most of the because in our cell optimum temperature is 37. So it is where the optimum capacity of the DNA polymerase enzyme is there. But nowadays this DNA polymerase enzyme which are used in the replication process, they don't use this eukaryotic cell. They use something called as an thermostable DNA polymerase enzyme. What is thermostable DNA polymerase enzyme? The DNA polymerase enzyme which are extracted from the bacteria which are living in a hot environment, especially hot water, something like that, where the temperature is around 65 or 70-degree temperature. Because these bacteria are naturally living in this hot temperature, their DNA polymerase enzyme, right, they are also having optimum temperature around 60 to 65 or more than that. So what happens is that once your denaturation is over, annealing step is over, we don't require to go up to 40-degree temperature to extension process. Extension process can also start at around 65 or 70 depending on which enzyme which are using, and entire process become speedy, and that is what we call something known as an thermostable DNA polymerase enzyme. So let us summarize the process. What is the DNA? What is the process? So what is the requirement in the DNA? So basically the entire process is performed by thermal cycling, 90-degree, 65-degree. If it's a regular enzyme, then we have to go 40-degree, and if it's thermostable, then we can perform at 65 also. And again we have to get back to 90-degree, right? So denaturation, annealing and elongation. So what are the requirement we required? First, DNA template. The DNA which is required, it's a target DNA which is required to amplify. Then obviously we required enzyme, the main hero of our procedure, right? DNA polymerase enzyme. Then we required primer. So here, as I have explained, primers are nothing but the starting point. Naturally it's a RNA primer, but in a PCR process you can use primer. The only purpose is to have a starting point, because DNA polymerase enzyme cannot recognize the single strand DNA. Then what you required? Obviously we required nucleotide molecules which is expected to join the strand. So we required nucleotide molecules, and then we required the buffer solution as a medium to have the reaction. So this is the PCR process. Basically it's a in vitro process happening within the happening outside of the cell and making lots of copies of the DNA. So third things that is to be discussed is about the viruses. If you see the paper, in all the papers which I have analyzed, 2 or 3 papers which are generally asked from the virus or which are related to virus. So generally for the virus, what I have broadly analyzed is that 3 things about the viruses are important to understand. First thing is that what is the basically viruses? Uh, Second thing, how virus replicates, how they work. And third thing is about the third thing about the virus is that what kind of the genetic material it is having. So let us see viruses between. So I hope all you know that viruses are basically considered as a link between living and non-living. What we mean by link between living and non-living is that viruses are what we call is a living. If we see the term living, generally in a biology, the term living means something which is metabolically active, right? Something or some organism, if they have active metabolism, then we call as a living. So what is the key unique feature of the virus is that when viruses are outside of something called suitable host, right? What is suitable host? I think we have discussed in our previous lecture, right? What we discuss is that when viruses infects the bacteria, it inside the bacteria, right? It uses the machinery which the bacteria is having and replicate itself. So what is basically viruses are doing? Whenever they are outside of the living cell, they are not doing any metabolic activity. Outside, they are as good as dead. But once they found some suitable host, they can get inside. Once they get inside, they will start the activity mainly related to replication of their genetic material. So what any organism wants, right? Any organism, the purpose is to have a replication of the genetic material and they want to spread as much as possible. So when viruses comes in a contact with the suitable host, they will start to replicate their genetic material. So we can say when it is inside the suitable host, it is living. But when it is outside, it is not doing any replication, any metabolic activity. That is why that's a non-living. So basically it's a link between living and non-living. Second thing about the genetic the virus is that as we know, most of all the prokaryotic cell or the eukaryotic cell we have discussed, we know that all this, no matter it's a bacteria or a advanced eukaryotic cell, they all are containing DNA as their genetic material. Now, the reason is the DNA is the genetic material is that it's a much more stable molecule. If you remember last time we have discussed that DNA is a much more stable against hydrolysis because it is not having any free hydroxyl group for hydrolyzation. So DNA is a much more stable molecule and that is why all the organism has a DNA as the genetic material. But that is not case with the viruses. Viruses can have either DNA or either RNA as their genetic material. Now the question can be that is that how RNA is possible to have a genetic material, right? But to the surprise, many scientists firmly believe that initially when organisms started to appear on the earth, they were containing RNA as their genetic material, not DNA. Now reason, let me explain. If you remember our DNA replication process that we have discussed, right? In that we have discussed that when the DNA replication process the starting of the replication process required RNA primer. We required RNA primer because the DNA polymerase enzyme, which is responsible for replication process, cannot read the single strand. And that is why it requires RNA primer as a starting point to start the replication process. So in short, we can say if DNA wants to replicate, they required help from RNA to start replication process. So DNA cannot replicate itself. They required help from RNA for the replication process. But on contrary, if you discuss about the RNA, so we know if we want to convert DNA, the DNA replication process, in short we can call DNA to DNA, right? It's a DNA to DNA. So if we want to create a new DNA from the old one, we require DNA polymerase enzyme. That's the main enzyme which can read it and add the nucleotide base molecule. Same way, if we want to convert RNA into RNA, we want to make the copy of RNA that don't require help of any strand. The enzyme which can form RNA to RNA, it is known as a RNA dependent RNA polymerase. RNA dependent RNA polymerase enzyme. And we know RNA is a single strand. So when this RNA polymerase enzyme required to work, they can read a single strand and they can form the copy of it. They don't require help of any DNA or any other extra molecules to start replication process. And that is why RNA can replicate itself without any help. So DNA required help of the RNA, but that is not case with the RNA. And that is why many scientists believe that it was the RNA was the genetic material initially. So what happened is that viruses can have RNA as their genetic material. It is possible in a virus to have a RNA as the genetic material. So before we start, let us briefly discuss this process. I think that might also be the useful in examination, something called basic dogma, right? So basic dogma of life or basic process in life. So as we know, enzyme and the processes. So DNA to DNA, this process is known as a replication process, right? And the enzyme which participate in is DNA polymerase enzyme. Now second process, we take DNA as our starting material and form RNA. That is known as a transcription process, right? We all know transcription process. And which enzyme perform the transcription process? It is known as a RNA polymerase. That is also we know, right? RNA polymerase enzyme. But remember, RNA polymerase enzyme don't require starting material. They can directly bind with the single strand DNA, they can form it. So it is known as a RNA polymerase. And because here the starting material is RNA, this RNA polymerase is also known as a DNA dependent RNA polymerase. So it's a DNA dependent RNA polymerase which performs this transcription process and converts DNA into RNA. Now third process is that it's conversion of RNA to RNA. Now where this RNA to RNA is performed, I will explain in subsequent part. So we take the strand of the RNA and make the complementary strand. And the enzyme which is responsible for it is also, because it is polymerizing RNA, it is known as a RNA polymerase. But because here starting material is RNA, it is known as a RNA dependent RNA polymerase. So RNA dependent RNA polymerase convert one strand of the RNA and copy and make the complementary strand. And fourth and the last one, we also have some process is called as a starting material is RNA and convert into DNA. So because the transcription is DNA to RNA, reverse conversion from RNA to DNA is known as a reverse transcription. And the enzyme which is responsible for is known as a reverse transcriptase enzyme. So main four processes: DNA to DNA, replication, DNA polymerase; DNA to RNA, transcription, DNA dependent RNA polymerase; then RNA to RNA, RNA dependent RNA polymerase; and RNA to DNA, it is reverse transcriptase process. Now where all this process is applicable, these all processes are applicable when it's come to viral replication. So basically what happened with the virus is that virus is basically when they enter into suitable host. So this is the suitable host, bacteria you can imagine, and this is the virus. So when they infect the bacteria, there are two possibilities for the bacteria. And what are the possibilities? These possibilities are known as the two phases. Either it is known as a lytic phase or lysogenic phase. So let us see what is lytic phase and lysogenic phase. So what happened, as you see in this figure, that once the virus infects the bacteria, so you can see this is virus, bacteria. So now this genetic material of the virus has entered into the bacteria. Now what happens, that once this is entered into the bacteria, this viral genome, it will try to take all the machinery that bacteria is having for its own replication. Ideally, bacteria is having DNA polymerase enzyme, nucleotide, everything, because they want to replicate their genetic material. So now what this viral DNA will do, it will hijack all this machinery, and rather than allowing this bacteria to replicate, they will use this machinery in replicating their own genetic sequence. So you can see this started to replicate their own genetic sequence. Now once they start lots of copies of the DNA. Then the second step starts. So once your replication is over, then transcription and translation start. So because this DNA, basically they are forming these kind of the cell structure. So you can see once the lots of copies has been formed, then they will start to make lots of copy of this head and this tail. So you can see second step, they will start to make lots of copies. And once we have lots of copies of head and genetic material, they will assemble and they will create the complete virus. Now once all this energy that this cell is having, or all the nutrition that this cell can offer to this viral has been exhausted, everything is utilized, now this cell has no more energy. So now what happens, once everything is over, the cell will lyse and all the new viruses will come out. And because in this phase where virus enters the bacteria or a host cell and ultimately kills the host cell, this is known as a lytic cycle. So lytic cycle is where the virus invades the suitable host and ultimately kills it. Now there is also second possibility. Ultimately what this virus wants is that replication of the genetic material. So second option what they can choose is that rather than having this violent way to replicate, what they will do by non-violent way, this viral DNA simply merges with the bacterial DNA. So you can see it's simple merge with the bacterial DNA. Now the same thing, whenever bacteria wants to divide, they have to replicate the entire genetic sequence. So once they copy their own genetic material, along with that they will also copy the viral DNA. So you can see all the subsequent progenies are having viral DNA at their structure. Here also the purpose is solved. Purpose is to have a maximum copy of the DNA. So you can see that is also solved. And this phase where the viral DNA integrates into host DNA, that are known as a lysogenic phase. So lysogenic phase is a phase where your viral DNA enters into the host phase. So either whenever virus infects the suitable host, either it go lytic phase or either it go lysogenic phase. Now what is our question? Our question is that the DNA can have, or viruses can have two genetic material, two kind of the genetic material. Either it can have a DNA or either it can have a RNA. Now DNA, that is in that also possible, either virus can have a single strand of the DNA or either it can have a double strand of the DNA, right? Because DNA can be double stranded. But virus, or when it comes to RNA, RNA is only single strand. So one of the way to classify the viruses is that what are the genetic material they are having. And the questions which are asked in examination is that they will give you the example of the virus and they can ask you that whether this is a DNA virus or RNA virus. Right. In RNA also, obviously if your virus is containing RNA and when it infects the host cell, first what is they required to do is that they required to convert this RNA into DNA, because the replication is only possible with the DNA. So what this virus required to do is that they required enzyme called reverse transcriptase. So what they do, they will use the reverse transcriptase and this RNA first convert into DNA and then the same process start. So reverse transcriptase is the enzyme which converts RNA into DNA. Now in RNA also, if you see the example of the virus, this when it contains RNA, generally this RNA comes with some sign. Either it will say something like this, it's a minus single strand RNA virus, or it's a positive single strand RNA virus. Let us take example, then it will make clear. So you can see, as you can see, this is list of some viruses, and generally the question which are asked is that they will give you disease and they will ask that which viruses are causing this disease. So obviously the or question can be asked like this, that hepatitis B is a what kind of the virus? Either it contains single strand DNA, either it contains single strand RNA, or double strand DNA. So that's why I was explaining that what is double strand DNA, single strand DNA, or the single strand RNA virus. Now if you see this example, these are the examples of some of the viruses which are causing some disease. So obviously it is not possible that we can remember all the virus in the whether they are DNA or RNA type and what disease it is causing. So what generally the trends from the examiner or the this examination body is that they will ask you questions about some of the prominent diseases which are very prevalent in the either which are important from the Indian perspective. Some diseases are very high in Indian population, or which are new disease. For example, one of the disease where Indian government is having also making lots of efforts is the HIV, right? So HIV is there, then influenza virus is there, then polio viruses are there, rabies viruses are there. So these viruses are in a lot more common discussion compared to some rare viruses. So it's not, as I said, it's not possible to remember all viruses, but at least the viruses or the disease which are caused by the viruses where Indian government is giving emphasis, that viruses you require to remember, and what kind of the viral type is that. So if you see in this all this virus, they have given the name of the disease, which virus are causing this, and also whether this is a single strand or a double strand. So let us take the example of adenovirus, right? Adenovirus causes respiratory tract infection. It's a double strand DNA. Its genetic is double strand DNA. But contrary to that, you can see the HIV virus. So HIV, which causes AIDS, it's a double strand RNA virus. So it's a double strand RNA. Single strand RNA. It's a SS for single strand, right? So it's my mistake. So it's a single strand RNA. But you can see whenever there is RNA, it comes with the sign minus or plus. So I don't know if it is asked, but what is basically this minus in plus is that, as you know, when the RNA replication, if you remember the transcription process, where the DNA is being used as a strand and the RNA is formed. So whenever RNA is formed, there is something called sense strand and there is something called anti-sense strand. So when two strands are joined, when the RNA is from the DNA, RNA polymerase enzyme responsible for making RNA from DNA, right? So DNA converts into RNA, right? So here is the RNA. So when this kind of the process starts, the strand on which this RNA polymerase is working, it is known as an anti-sense strand. And the opposite strand is known as a sense strand. So what is anti-sense and sense strand? You can look at in your book. I am not explaining in a detail because that will take another half an hour too. So basically what happens, if your DNA is directly acting, the strand which is made, it is an anti-sense strand, and opposite strand is the known as a sense strand, and where it is working, it is an anti-sense strand. Same way, when virus is containing the genetic material, when it's containing RNA as the genetic material, if this RNA is directly converted into DNA without any process, it's known as a plus sense strand. But if this RNA, the viral RNA, first converted into complementary strand, and then this complementary strand is converted into DNA, that is known as a minus single strand RNA. So we know if we want to convert directly RNA to DNA, the enzyme required is reverse transcriptase. So when RNA is converted into another RNA, the enzyme required is RNA dependent RNA polymerase. And then again, because we have this RNA, it converts into DNA by reverse transcriptase. So if the RNA is ready-made is used to convert into DNA, these viruses are known as that viruses are denoted as a plus sign. So you can see all this plus. Plus means this strand is ready-made available to copy. this minus means first this RNA required to convert into complementary strand and then it is converted into DNA. So here these examples, I have taken few examples, but as I have said, it's not possible to remember all. What you require is the prominent disease. The disease which are frequently, or the disease which are prominent in India. For example, dengue, HIV, then obviously right now coronavirus, which is also the RNA virus, then influenza viruses, at least the disease against which Indian government is supplying the vaccine in child or in adult. Whatever vaccines are available, at least that viral, and along with that, whether it's a DNA or RNA virus, that is required to be used is you have to remember. Now the next question, which is also related with this, when the virus infect the bacteria, is that it's one of the way of genetic transfer. So what is genetic transfer? When the DNA material from one organism is transferred to another organism, that is known as a genetic material transfer. Now this genetic material transfer can be performed in a three way: transformation, conjugation and transduction. Basically it's the process of transferring the genetic material from one organism to another organism, and it can proceed by three way: transformation, conjugation and transduction. So let us see what is transformation. Transformation is a naked DNA transfer. What happened? One organism dies, its genetic material is released in environment, and by chance it reaches to another organism, where this second organism takes up this genetic material and integrated into. Yes, this is a chance process, right? It's not happening every time, it's a chance process. So the transfer of the genetic material from one organism to another organism in an open environment, that is known as a transformation process. Second is the conjugation process. In a conjugation process, what generally happens? It happens between two bacteria. So here, as you remember in our last discussion, bacteria can have a plasmids. It's the extra chromosomal DNA which can replicate on its own. So what happens is that when this plasmid is to be transferred, this kind of the bridge is formed between two bacteria. It is known as a phylus, or it is also known as a sex bridge. So now what happens? This plasmid replicates itself, it creates the copy, and this copy is transferred to another bacteria. So here the DNA which is transferred by using this kind of the bridge like structure, sex bridge or phylus like structure, that is known as a conjugation process. So one way to transfer is the transformation. It is characterized by naked DNA transfer, just like that. Where you require a special bridge, that is conjugation. And third, where viruses can act as a carrier and transfer the genetic material from one to other, that is known as a transduction process. So you can see transduction, what is basically happening? Virus are entering into the DNA, and we know when virus enter into the bacteria, what happens? Ultimately it kills the bacteria, right? Lytic phase. So once the bacteria is died, this viral DNA, it's packed into bacterial DNA, packed into viral, and then it transferred the other bacteria. So this kind of the transformation, where the virus is a mean, virus is transferring from one to another bacteria, these are known as a transduction. So question was that virus mediated transfer of host DNA from one cell to another cell is known as a transduction process. Then another question was, it was related to hybridoma technique. So we all know what is hybridoma technique. Basically it is the process by which we form monoclonal antibodies. Monoclonal antibodies are formed due to fusion between beta cell and the myeloma cell. Beta cell, so let us see, beta cell basically what they do, spleen cell or beta cell, it produces the antibody. Myeloma cell, basically they are B cell only, but the key characteristic of this myeloma cell is that they divide very rapidly, continuously. And the cell which is formed between the conjugation or between the hybridization between myeloma and spleen cell are known as a hybridoma cell. So hybridoma cell has a two characteristic. First, it divides rapidly, and second thing is that it also produces the antibody. Now, once you have this division of the cell, when it comes to selection, the media which is used is known as a HAT media. So question was that in a hybridoma technique, so what is hybridoma technique? Is the process by which B cell, which is producing antibody, is merged or it is linked to myeloma cell which divide rapidly to create the monoclonal antibodies, and these cells are screened by media called HAT media. So HAT stood for hypoxanthine aminopterin thymidine media. So how this media work? So basically, if you remember, the DNA can be formed by two methods. One is known as a salvage pathway, and second is known as a de novo pathway. So de novo pathway is completely new, right? Nucleotide. If we talk about the how purine and pyrimidine nitrogen bases are synthesized, they are synthesized by either salvage pathway or de novo pathway. Now exactly we are not going in a detail because that is the beyond the scope of this discussion, but I am giving you just a brief idea. So what happens when you use aminopterin in this medium? It will block this de novo pathway. So your DNA will not synthesize, nucleotide will not synthesize by de novo pathway. The only way it can synthesize is by salvage pathway. So aminopterin, the function is to stop the formation of nucleotide by de novo synthesis. Now the only pathway which is open is the salvage pathway. So what happens in a salvage pathway? In a salvage pathway, okay, so aminopterin basically it's a substrate for phosphoribosylate. It inhibits the tetrahydrofolate reductase, and it stops the synthesis of adenine, guanine, thymine, and cytosine. So they will not form by de novo pathway. Now another two component of the ingredient, hypoxanthine. What this hypoxanthine do? Hypoxanthine, when it react with enzyme called hypoxanthine guanine phosphoribosyl transferase, it convert into adenine and guanine. So you have adenine and guanine shortage, and thiamidine converted into thiamine. So because aminopterin, it stops the synthesis of adenine, guanine, thymine, and cytosine. Sorry, not adenine, guanine, and thymine. We required some two extra ingredient which can compensate this loss. So hypoxanthine, it fill up the loss for adenine and guanine, and thiamidine converted into thiamine. And this is how the cell is screened, right? So we are, I am not explaining exactly how it is stopping the tetrahydrofolate is used. If anyone is interested, you can go to Satyanaran. They have given this description, and you can read about it. So that's it from my side. Thank you everyone for the patient listening, and please share your valuable feedback. If you think that some topics I have not described properly, or in some points if I have erred, right? I am a human, so it's possible that I have erred somewhere. So it is the case. Please share your feedback on this email ID. Whatever things that you think that I require to improve, I require to make my lecture more cogent, attractive way. So please share your feedback, and thank you very much.